Good morning to all. Hello friends. I am your friend Dr. Shubh Patel. Today we will discuss about a question from Lab Animal Medicine. Question. In which of the following species are females considered spontaneous ovulators? Meadow vole, ferret, gerbil, degu, ground squirrel. Answer, C. gerbil. The question asks about spontaneous ovulators. To understand this well, we must first understand what spontaneous ovulation means, and how it differs from induced ovulation. In spontaneous ovulation, the female produces and releases eggs cyclically without needing mating stimulation. Hormonal changes, especially involving estrogen, naturally lead to the release of an egg during the estrus or menstrual cycle. In contrast, in induced ovulation, mating triggers the release of an egg. Without mating, ovulation does not happen. Now, let's go through each species given in the question to understand which fits the pattern of spontaneous ovulation. First option, meadow vole. Meadow voles are small rodents found in North America, commonly living in grasslands and meadows. They belong to the microtine rodents, and interestingly, they are classified as induced ovulators. In their reproductive biology, physical stimulation from mating is necessary to trigger the hormonal cascade leading to ovulation. Without mating, ovulation will not occur in these animals. This means that meadow voles are not spontaneous ovulators, and so this is an incorrect answer. Second option, ferret. Ferrets are well known in reproductive physiology studies because they are classic examples of induced ovulators. In female ferrets, mating triggers a surge in luteinizing hormone, LH, which then leads to ovulation. If the female does not mate, she remains in prolonged estrus, which can even become dangerous and lead to health complications like persistent hyperestrogenism and subsequent aplastic anemia. Therefore, ferrets are not spontaneous ovulators, and this is another incorrect option. Third option, gerbil. Now we come to the gerbil, specifically the Mongolian gerbil, Merians unguiculatus, which is commonly used in laboratory research. Gerbils are, importantly, spontaneous ovulators. They have regular estrus cycles, typically about every 4 to 6 days, and ovulate irrespective of mating activity. Hormonal regulation governs their cycle. Estrogen levels rise naturally, triggering the pre-ovulatory surge of luteinizing hormone, which results in ovulation. This fits the definition perfectly and makes the gerbil the correct answer. Additionally, from a research perspective, this characteristic is helpful when designing breeding programs because ovulation does not depend on male presence. Fourth option, degu. Degus, Octodon degus, are South American rodents, social animals often used in behavioral and neurobiological studies. Interestingly, degus are seasonal breeders and have characteristics of spontaneous ovulators, but in a seasonal context. However, it is not regular spontaneous ovulation throughout the year like the gerbil. Degus have regulated breeding seasons, and their ovulation happens spontaneously only during these windows, primarily influenced by photoperiod, light exposure. Since the question does not specify seasonality, and we're looking for a general spontaneous ovulator, the degu does not fully qualify in this context. Therefore, this is not the correct answer. Fifth option, ground squirrel. Ground squirrels are members of the Sciuridae family, and they are typically induced ovulators. Mating stimulates ovulation in these animals. Also, they are seasonal breeders, with reproductive activity restricted to specific times of the year. Their ovulatory process is triggered primarily by copulation rather than occurring on a regular hormonal cycle. Thus, ground squirrels are not spontaneous ovulators, making this an incorrect option. Conclusion of species discussion. Out of all the options, only the gerbil exhibits regular, hormonally driven spontaneous ovulation, making it the correct answer. Now, let's expand with additional context so you can understand this deeply and remember it well. Additional explanation, spontaneous versus induced ovulation facts and figures. Spontaneous ovulators rely on cyclic hormonal changes. Estrogen levels rise naturally during the follicular phase, triggering the hypothalamus and pituitary gland to release luteinizing hormone, LH, leading to ovulation. Examples of spontaneous ovulators include humans, primates, guinea pigs, mice, rats, cows, and gerbils. Induced ovulators require mating stimulus for ovulation. This mechanical or neuroendocrine stimulation during copulation triggers the hypothalamus to release GnRH, gonadotropin-releasing hormone, which leads to the LH surge necessary for ovulation. Examples include cats, rabbits, ferrets, camels, and ground squirrels. Some species show seasonal spontaneous ovulation, like sheep and deer, where environmental factors such as day length trigger the cycle, but ovulation within the season is spontaneous. Frequency and cycle length. Gerbils, estrus cycle of approximately 4 to 6 days. Mice, 4 to 5 days. Rats, 4 to 5 days. Ferrets, prolonged estrus without mating. Rabbits, continuous estrus until mating-induced ovulation. Refer comparative table, ovulation types in lab and research animals. Fun facts and figures about reproductive strategies. In spontaneous ovulators, even if mating does not occur, ovulation and hormonal cycles continue. This is why, for example, unbred female mice still display estrus cycles. In induced ovulators, the strategy conserves energy by preventing unnecessary ovulation. Since mating triggers ovulation, it synchronizes gamete availability. Fertilization rates in induced ovulators can be very high because ovulation is closely linked to mating. Laboratory breeding colonies prefer spontaneous ovulators for ease of managing breeding programs. Connection to placenta types, as requested. Spontaneous ovulators like gerbils, rats, and mice also tend to have hemachorial placentas where the maternal blood is in direct contact with the chorion. This supports efficient nutrient transfer for fast-growing embryos. Induced ovulators such as rabbits and cats also often have hemachorial or endotheliachorial placentation, reflecting their need for efficient fetal development once ovulation and fertilization have occurred. Closing summary. The correct answer to the question is gerbil, as it is a true spontaneous ovulator with regular estrus cycles independent of mating stimulation. Other species listed are either induced ovulators, like ferrets and ground squirrels, or have seasonal spontaneous cycles, like the degu. This understanding is crucial not only for exam answers but also for practical breeding management in laboratory animal colonies and for appreciating evolutionary reproductive strategies across mammals. Let's go step by step, like a full revision lesson. 1. Practice MCQs, with explanation for each answer. Question 1. Which of the following species is an induced ovulator? A. Rabbit. B. Guinea pig. C. Rat. D. Mouse. Answer: A. Rabbit. Explanation: rabbits are classic induced ovulators. Mating stimulates ovulation. Guinea pig, rat, and mouse are all spontaneous ovulators. Question 2. In spontaneous ovulators, what primarily triggers the ovulation process? A. Mating stimulation. B. Seasonal light exposure. C. Hormonal cycle changes. D. Temperature fluctuations. Answer: C. Hormonal cycle changes. Explanation: in spontaneous ovulators, internal hormonal cues, like estrogen rise triggering LH surge, drive ovulation, independent of external mating stimuli. Question 3. Which of the following is a spontaneous ovulator? A. Ferret. B. Cat. C. Gerbil. D. Ground squirrel. Answer: C. Gerbil. Explanation: ferret, cat, and ground squirrel are induced ovulators. Gerbil is a spontaneous ovulator. Question 4. Which species exhibits seasonal spontaneous ovulation? A. Sheep. B. Gerbil. C. Rabbit. D. Rat. Answer: A. Sheep. Explanation: sheep are seasonal breeders but ovulate spontaneously within the breeding season. Question 5. Which of the following best describes the reproductive cycle of the ferret? A. Continuous estrus, spontaneous ovulation. B. Seasonal breeder, induced ovulation. C. Seasonal breeder, spontaneous ovulation. D. Prolonged estrus, induced ovulation. Answer: D. Prolonged estrus, induced ovulation. Explanation: ferrets remain in estrus until mating occurs, which then induces ovulation. 2. Memory trick to instantly recall ovulation types. Let's make this easy to remember. Trick. Spont means it happens on its own. Induced needs an external push. Spontaneous ovulators. Think of routine clockwork. Examples: mouse, rat, gerbil, guinea pig, sheep, cow, human. Induced ovulators. Think of trigger required. Examples: rabbit, ferret, cat, camel, ground squirrel. 3 extra detail: reproductive hormones and ovulation trigger. To truly master this, you need to understand the hormonal control. Spontaneous ovulators. Regulated by internal hormonal cycles. Estrogen rises as follicles mature. High estrogen triggers the hypothalamus to release GnRH, gonadotropin-releasing hormone. GnRH stimulates the pituitary gland to release LH, luteinizing hormone. LH surge causes ovulation. Mating is optional even if no mating occurs, the cycle progresses naturally. Example: in gerbils, the estrus cycle is about 4 to 6 days, with clear hormonal peaks leading to ovulation. Induced ovulators. No spontaneous hormone surge. Copulation stimulates sensory nerves in the reproductive tract. Neural signal goes to the hypothalamus. Hypothalamus releases GnRH. Pituitary gland releases LH, causing ovulation. Without mating, no ovulation happens. Example: in rabbits, ovulation happens approximately 10 hours after mating. Extra insights. Prolactin and progesterone increase after ovulation, supporting pregnancy if fertilization occurs. In many spontaneous ovulators, external factors like light, photoperiod, or social cues can slightly influence cycle timing, but they are not essential for ovulation. Induced ovulation often evolves in species with solitary or unpredictable mating encounters, to increase reproductive efficiency. Final recap summary. The question asked for a spontaneous ovulator. Only gerbil fits has regular estrus cycles independent of mating. Others, ferret, ground squirrel, are induced ovulators. Spontaneous ovulation is hormonally driven internally. Induced ovulation requires mating stimulation to initiate ovulation. Hormones involved: GnRH rightward arrow, LH surge rightward arrow, ovulation. Today, we already discussed spontaneous and induced ovulation, and you've understood that gerbil is a spontaneous ovulator, while ferret and ground squirrel are induced ovulators. But I want you to understand this not just as a fact, but as a biological story, how evolution, reproductive strategies, and environmental needs shape these patterns. Let's first talk about gestation length and litter size. You might wonder, do spontaneous and induced ovulators differ in how long their pregnancies are, or how many offspring they have? The answer is, yes, they generally do, and the reasons are very logical. Spontaneous ovulators, like gerbils, rats, and mice, have short gestation periods and moderate to large litter sizes. Think of the gerbil. It has a gestation period of about 24 to 26 days, and typically produces 4 to 6 pups in a litter, sometimes even more. The rat and mouse are similar, with gestation around 20 to 22 days, and litters ranging from 6 to 12 pups. Why is this? Because these animals are prey species. Their survival depends on reproductive efficiency. They breed quickly and often, so spontaneous ovulation ensures that whether a male is nearby or not, their body is prepared to ovulate, mate, and reproduce at regular intervals. Now compare this to induced ovulators. Take the ferret for example. Ferrets have a longer gestation of about 41 to 42 days, and their litter size is usually 6 to 9 kits. Interestingly, although they have a good number of offspring, they don't ovulate unless they mate. This is an energy-saving strategy because if there is no male, there's no need to prepare for pregnancy. The rabbit, another classic induced ovulator, has a gestation period of 30 to 32 days, with litter sizes averaging 4 to 12 kits. Rabbits also use this efficient system of only ovulating when mating occurs, ensuring that every ovulated egg has a good chance of being fertilized. Ground squirrels, also induced ovulators, have seasonal breeding, with gestation periods around 28 to 30 days and litter sizes of 4 to 8 pups. Here, seasonality also plays a role, synchronizing birth with food availability. So, you see, gestation length and litter size tie in closely with ovulation type, species ecology, and environmental adaptation. Now let's move to the reproductive parameters for common laboratory animals, but I want to explain this as if I am giving you an internal picture of each species. Take the mouse, for example. Mice are spontaneous ovulators with estrous cycles of about 4 to 5 days. They reach puberty at around 6 weeks of age. Their mating is very prolific. If you put a male and female together, chances are they will mate quickly, and since females ovulate spontaneously, timing is rarely an issue. Rats behave similarly. They have an estrous cycle of about 4 to 5 days, spontaneous ovulation, and gestation of about 21 to 23 days. Their litter size is often 8 to 12 pups, making them ideal models for reproductive and developmental studies. Now, gerbils, our correct answer today, are fascinating. Their estrous cycle is slightly longer, about 4 to 6 days, and they reach puberty at around 65 to 85 days of age. They ovulate spontaneously, gestation lasts around 24 to 26 days, and they typically have litters of 4 to 7 pups. Contrast this with ferrets. Ferrets, being induced ovulators, remain in estrus until mating. They don't follow a regular cycle like rats or mice. If mating doesn't occur, they can suffer from prolonged estrus, which can be dangerous. Their gestation lasts about 42 days, and litter sizes range from 6 to 9 kits. Finally, let's think about the guinea pig. Although not in our question today, it is an important lab animal. Guinea pigs are spontaneous ovulators with longer gestation periods of about 59 to 72 days, and litters of 2 to 4 pups, larger pups at birth compared to other rodents. So, here's what you should take away. Spontaneous ovulators tend to have regular, predictable reproductive cycles, while induced ovulators rely on mating to trigger reproduction, conserving energy when males are not present. Alright. Let's begin by understanding the basics of reproductive hormone regulation, and then I will explain both spontaneous ovulation and induced ovulation, and how their feedback loops differ. Hormonal control of reproduction is like a finely tuned orchestra. The brain, the pituitary gland, and the ovaries all play their parts in harmony. The two main players in this system are the hypothalamus and the pituitary gland, which send hormonal signals to the ovaries to control the growth and release of eggs. The key hormones we will focus on are GnRH, gonadotropin-releasing hormone, produced by the hypothalamus. FSH, follicle-stimulating hormone, from the pituitary. LH, luteinizing hormone, from the pituitary. Estrogen, produced by the developing ovarian follicles. Progesterone, produced by the corpus luteum after ovulation. Now let's walk through the two systems, as if they are two different stories. Story 1. The spontaneous ovulator. Let's imagine we are observing a gerbil, since that was our correct answer. In the spontaneous ovulator, the reproductive cycle runs on its own internal clock, independent of external stimuli. It starts in the hypothalamus, which regularly releases pulses of GnRH. This GnRH acts on the pituitary gland, which then secretes FSH and LH into the bloodstream. FSH travels to the ovaries and stimulates the growth and development of follicles, little sacs in the ovary that contain the eggs. As these follicles grow, they start producing increasing amounts of estrogen. Estrogen has a fascinating dual role. In the early phase, it feeds back negatively to the hypothalamus and pituitary to keep FSH and LH levels moderate. But as estrogen levels rise further, especially as the dominant follicle matures, this feedback flips to positive feedback. At a critical threshold of high estrogen concentration, the positive feedback loop kicks in. This triggers the hypothalamus to release a surge of GnRH, and the pituitary responds with a surge of LH, and to a lesser extent, FSH. This LH surge is what causes the mature follicle to rupture and release the egg ovulation. After ovulation, the ruptured follicle transforms into the corpus luteum, which produces progesterone to prepare the uterus for pregnancy. If pregnancy does not occur, the corpus luteum regresses, progesterone drops, and the cycle begins again. This cycle continues regularly, every few days in small rodents like the gerbil and mouse, independent of mating. Even if no male is present, the hormones will still cycle, and ovulation will occur naturally. This is why it is called spontaneous ovulation. The internal hormonal environment alone is enough to drive the process. Story 2. The induced ovulator. Now, let's imagine we are observing a ferret or a rabbit. Things work a bit differently here. In induced ovulators, the early part of the cycle looks somewhat similar. The hypothalamus releases GnRH in low, basal amounts, the pituitary releases low levels of FSH, and follicles start to develop. Estrogen is produced by these growing follicles, but without external stimulation, estrogen levels stay moderate and do not reach the threshold to flip the feedback loop from negative to positive. The female remains in a state called prolonged estrus. She is hormonally ready, but without a mating stimulus, the system does not progress to ovulation. Now, here is the key moment: mating happens. During copulation, mechanical stimulation of the cervix and vaginal wall triggers neural signals that travel to the brain, specifically the hypothalamus. The hypothalamus responds by releasing a large surge of GnRH, which in turn prompts the pituitary gland to release a surge of LH. This LH surge, just like in spontaneous ovulators, leads to ovulation, the release of the mature egg from the follicle. After ovulation, the corpus luteum forms, producing progesterone to support pregnancy if fertilization occurs. But here's the crucial difference: without mating, there is no LH surge, and ovulation simply does not happen. The follicles may continue to produce estrogen for some time, keeping the female in estrus, but without that physical stimulus, the system remains idle. This is an energy-saving design. For animals like ferrets and rabbits, who may not encounter mates frequently, it avoids wasting energy on unnecessary ovulations. Comparing the two systems together. So you see, both systems use the same hormonal players, but the triggering mechanism is different. In spontaneous ovulators, the hormonal system drives itself on an internal schedule, with rising estrogen levels eventually prompting ovulation. In induced ovulators, the hormones remain in a state of readiness, waiting for external physical stimulation from mating to activate the final surge of GnRH and LH needed for ovulation. Both systems are elegant, adapted to the reproductive strategies and environmental conditions of the species. Final thought to anchor your memory. If you remember this simple logic, it becomes very natural. In spontaneous ovulators, estrogen builds up naturally, like water rising in a cup until it spills over. In induced ovulators, the cup is waiting, but it only fills and spills when mating physically turns on the faucet. Thank you for listening. Have a nice day ahead and save your patient with love and dedication. Please subscribe and like.